The next few lectures in this series need some context from the tree of life. Now, we can't draw the whole tree. I don't know if anyone can, but here's our branch somewhere over here. The animals, or metazoa, if you'd rather. Our sister group, we already mentioned, the choanoflagellates. And there are some other flagellates in there. And then there are the fungi. The somewhat uncertain connection. We'll put in the amoebas over here. Now, there are lots of things called amoebas in the eukaryotic tree. Amoebas are a mode of motility, not a phylogenetic group. This group here that we share with the fungi and choanoflagellates and some smaller flagellate groups, these are the opisthokonts. Refers to the position of the flagellum. Opisthokonts have the flagellum, the cilium, at the back end of the cell. And when the flagellum or cilium beats, it propels the cell forward. Okay, now somewhere over here on the other side, another big familiar radiation, let's say that's the land plants. Land plants are part of the green algae, in effect. Of which there are very many groups. Off here at the edge, I'm going to put a little group of flagellates called the glaucophytes that most people have never heard of. And then somewhere over here, we'll put the red algae. The reason these are bunched together is that these are the eukaryotic organisms that originally established an endosymbiosis with some kind of cyanobacteria that became the chloroplasts, the plastids of today's photosynthetic eukaryotes. These three branches are the Archaeplastida, the ones that got plastids first. Now, it isn't entirely clear whether this was one acquisition of this symbiosis with the cyanobacterium or three. The pendulum has swung with recent articles, with recent investigations toward a single origin. There are a variety of other groups that we can put on this tree. Somewhere over here, we might put the Euglena type organisms, the Euglenozoa. Some things that are related to them. I am no doubt leaving off a great deal. And in the middle, I'm going to draw a large and rich group first. This is the Stramenophiles, which includes what we usually call brown algae, the kelps, for example. It also happens to include the diatoms, which is the first protist group we're going to consider. In here somewhere are the haptophytes. The alveolates, which includes dinoflagellates, ciliates, and a number of other interesting lineages. The rhizarians. And again, there are many things left out, but this group here, whatever its boundaries are, goes by the somewhat ungainly name, the SAR, capital, for Stramenophiles, alveolates, and rhizarians. Not very evocative, I know. Let's look at those plastids. We know the land plants are green, right? All the way down. They share their greenness with all the other green algal groups, of which, again, there is a great diversity. The glaucophytes, that means they're gray. But really they're sort of more blue-green. Those taking the lab will meet these glaucophytes, which you can now buy in culture from Carolina, of all things. They really look like they've swallowed a bacterium. They really look like they've got a blue-green alga in there. Then, of course, there's the red algae. But we know these are not the only photosynthetic organisms, okay? There are all kinds of other groups that acquired plastids from the Archaeplastida, acquired them secondarily or even tertiarily. One in particular that I'm going to squeeze in here one of my favorites, the cryptophytes, an ancient lineage of flagellates that somehow acquired their plastids. It's very clear that they got them from red algae. Many euglenozoans, not all, but many, are green. They got some plastids from green algae at some point. So did various kinds of amoebas. and some rhizarians, and probably some other things in there. In the process of acquiring plastids, many transformations might have taken place, many other inventions. The brown algae are brown because although they have plastids with chlorophyll in them, they also have accessory pigments. Brown chalk doesn't look great, so I'm going to use yellow instead. So diatoms and many other stramenophiles although not all, have a yellow or a brown plastid, as do haptophytes. It is not clear whether these things got their plastids secondarily in one event or many. It is clear that some subsequent transfers have gone on within this SAR clade. In fact, amongst the dinoflagellates, I'm going to depict one of these branches with all of these colors because some dinoflagellates appear to have serially replaced the plastid by successive tertiary endosymbiotic captures of the photosynthetic organelle. We'll cover that later. There are ciliates that have gone green. Let's put the ciliates in there. There are organisms within this group that have then secondarily lost plastids. I note it's not clear exactly what the relationship is, but that's a little confusing there. Let me fix that up. It's not clear what exactly the relationship is, but there are non-photosynthetic relatives of the cryptophytes called the catablepharids that live mostly by eating bacteria. They have either lost their photosynthetic organelles or they reflect the condition that the ancestors of the cryptophytes might have had. In contrast, there are very clearly some stramenophiles that have lost their plastids. A good example is the organisms called water molds that includes the blight that caused the famous Irish potato famine. Those are related to the diatoms and the kelps and things, but now live in a way much similar to fungi. So that's the context in which we're now going to discuss a series of eukaryotic groups which contrast informatively with the subjects we've been discussing in this lecture series: mitosis, the cell cycle, meiosis, motility in animals so far. So let's start with the diatoms here. Diatoms are all unicellular, although many are social, that is, live together, or colonial. They're all photosynthetic and they're encased in a silica exoskeleton. They come in two basic kinds. There's the centric diatoms, which in general are more or less radially arranged, where their structure is defined largely by their shell. And there's pennate diatoms, that is, ones that look like a pen, We're talking about a classic fountain pen here, which, had a nib and then a slit that conducts the ink. That slit is a structure called the raphe, a slit through the shell. So these are the pennate diatoms. Now I'm going to erase to continue here. The pennate diatoms include so-called araphid pennates, which I'll just draw sort of a random cell shape. And then there are various so-called multipolar diatoms. Whose shell architecture departs from the stereotype of the centrics. Who came first? The suggestion is that these multipolar forms branched off first, and that the relationship is something like this. Again, the centrics are organized around, their shells are organized on a sort of a radial plan, more or less like a petri dish. The pennates are much more recently diversified. Their shell also comes in two halves, as we'll see in a second, and they're mostly organized around this slit in the shell, the raphe. They all have a common structure to that shell, which is called the frustule. Sounds like it's going to be frustrating, but the frustule refers to a little bit cut off. It's not very flattering, is it? The frustule consists of two valves a series of what are called girdle bands. I'm drawing a kind of expanded view here. Now, this next set of girdle bands, these fit into the ones above. Bear with me here. As does this other valve. If you like, you can call these girdle bands, if you like fancier terms, you can call that a cingulum. Sounds like singular, but it's spelled with a C. One half of this assembly is the epitheca, and this is the epivalve. The other half, the smaller half that fits inside, is the hypotheca. and this is the hypovalve. Epi means upon, hypo is the opposite. That's what it looks like for a centric like this. Imagine turning it on its side. We've got sort of a petri dish assembly that's expansible based on the production of girdle bands. Similar thing for the penates, turning this diagram like that. The epivalve might look sort of like, oh, I don't know, a coffin lid, I guess. Perhaps some thin girdle bands there. Let's just draw a couple. So same idea, different shape, and with distinct polarity to it. So the diatoms are one of the, we commonly think that everything was invented before we came on the scene. By we, I mean animals. The diatoms are one of the groups that followed us onto the evolutionary scene within the eukaryotic tree of life. They are unknown before the Jurassic. And seeing as they're enclosed in a mineral shell, diatoms fossilize very well. You can, of course, buy diatomaceous earth. Their shells accumulated in great deposits in various places. And if you imagine that diatomaceous earth is composed of a bunch of crushed little glass microshells, which it is, one of the chief uses of it is to kill crawling insects in your household, or at least deter them, since the little bits of glass get into their breathing tubes. Okay, so they're unknown before the Jurassic. They proliferated in the Cretaceous, particularly the centrics, okay? And their arrival is correlated in general with global cooling. Making the cell wall out of silica, as they do, that is, out of glass, is probably a cheap strategy compared to making it out of organic material or, say, calcium carbonate. Why? Because, first of all, silica, silicon dioxide, is the raw material of planet Earth. That's what rocks are. Okay, they're making their shells out of rock. Diatoms take up silicic acid from the water and they precipitate it. It happens that it precipitates readily in acid. And it's deposited, in the case of the epivalve and the hypovalve, deposited as a single rock. In effect, they're marine grass. Grasses, too, silicify. And in fact, grasses also arose at about the same time and took over. Diatoms cannot live without their frustule. No one has ever, to my knowledge, made a diatom protoplast, although some species can live with a very thinly silicified frustule. Others, it's quite a heavy-duty structure. And the important thing about the frustule, okay, every invention that cells make to deal with their challenges is going to impose some constraints along with the opportunities that affords. One of the trades for this rocky shell is, first of all, diatoms don't have cilia. Same trade took place in the animal group, the arthropods. They gave up cilia for the sake of armor. The only diatom cells that have cilia are diatom sperm. ordinary vegetative cells can't swim. Again, since the frustula is basically a rock, it sinks. Dead diatoms sink to the bottom of the sea. In fact, live ones sink too, unless they do something about it or currents stir them up. But most important, the frustule constrains cell growth to one axis. Once made, these parts of the frustule can't expand this way. They can't balloon out. All they can do is expand, telescope out, and add more girdle bands. Within the frustule, if we look at it for sort of a stereotypical centric diatom structured like a petri dish, within the frustule, if this is the epivalve, and here's the hypovalve, I'll leave out the girdle bands for now. The cell membrane is right under the shell, but the cell structure within is typically dominated by a very extensive vacuole. Indeed, for a large centric diatom, most of the cell volume within the frustule might be vacuolar space. Why is that? So let's put a nucleus in here. And let's fit in plastids. In a typical centric diatom, there are many small plastids. Especially around the periphery, just beneath the shell. One way to rationalize this is to say a large fraction of diatom form is about spreading out these plastids, the photosynthetic organelles, to the greatest extent possible. So in a large species of diatom, those of you taking the lab course will likely see Cassina as an example, which might be hundreds of microns across. The skin containing all the plastids, basically the skin of the cell, is a very thin plastid-enriched layer connected by cytoplasmic tendrils around giant vacuolar spaces that are effectively dead. These spaces in here are created by the living cell, but are themselves non-living. There are various functions to vacuolar spaces, but let's stick for the moment just to taking up space. The vacuole is, as in land, is the similar organelle in land plants or red algae, basically a great big lysosome, the expanded version of the little bags of death wire. And incidentally, when you look at live diatoms, as with many other living cells, one of the ways to tell whether the space you're looking at is alive or dead is whether there's Brownian motion going on inside it. In dead spaces, little particles will be jittering around. In live spaces, they won't be. They're caged by macromolecular soup and cytoskeletal filaments and things like that. And indeed, running through many of these tendrils of cytoplasm, excuse me, that's supposed to go through there, you would find a microtubular cytoskeleton supporting traffic, including the traffic of plastids in and out along these cytoplasmic channels to the periphery. As lab students will see, many pennate diatoms likewise have a large vacuole with nucleus suspended in a kind of an island of cytoplasm in the center and maybe tendrils reaching out through there. But as it happens, very often have just two large plastids in there. And of course, many small diatoms might be small because the vacuole is a relatively small fraction of the internal volume, perhaps to the extent of hardly being there at all. Besides these stereotypical shapes, many have rather complex forms. A common feature is spines. For example, on forms like Chaetoceros, spines may be siliceous, that is, made of silica, or in some cases, chitinous, threads reaching out from the frustule, secreted through pores in the glass structure. And in some cases, there may be enough volume in extrusions of the frustule like this to even send out the solar panels to collect more rays. Chain formation is another common feature of diatoms. I mentioned that many are social or colonial. This Chaetoceros type are often found linked together in short chains. More or less in an orderly way. I'm drawing a pretty disorderly one here. Forms like these, chains and spines, we can look at as adaptations for a number of functions likely, but for one thing, it makes them a nasty sort of a mouthful, right? I mean, who wants to eat something shaped like that? With luck, those of you taking the lab will see someone who does. But so cell shape in diatoms and chain formation may be in part defensive, but it's also likely about remaining suspended in the water. Chains may be very complex in structure. Another common form that lab students will hopefully get to know. Makes interlinked glass struts looking like a pair of hands holding on to each other. And perhaps these two have spines sticking out. Aside from being interesting adaptations, structures like these also pose some interesting questions about morphogenesis. Some of the readings will concern things like that, such as how do they construct these things? How do they aim them? How do they link them up? How do they make all that glass? The shells of diatoms are among the finest glasswork the world has ever known. They are remarkable feats of cellular engineering, and how they are patterned and produced remains, as far as I know, largely mysterious. Cell division is also very different in diatoms compared to what you've heard of in animal cells. with one major exception, which we'll get to. To start with, diatoms don't have centrioles or centrosomes in the conventional sense, except the sperm have centrioles and flagella. They do, however, have a microtubule organizing center. That green thing there is called the microtubule center, creatively. It is an MTOC, that is, it organizes microtubules. It has no structural relationship with the centriole. In the electron microscope, it just looks like a dark round ball with microtubules emanating out of it. And traffic along those microtubules organizes cytoplasmic tendrils. Remember, this is vacuolar space. Here, there's the nucleus parked in this island of cytoplasm, kind of in the center, with the plastids out at the periphery where they can soak up the sunlight. Now, as these cells, and I'm drawing a pennate diatom here because much of what people have learned about mitosis in diatoms comes from pennate diatoms, particularly large ones, where the mitotic apparatus is relatively large and clearly visible in the light microscope. As these cells approach mitosis, the microtubule center grows a kind of a little stem on the side facing the nucleus. So here's the nuclear membrane. And the microtubule center with, again, microtubules emanating straight out from it. And that stem elongates into a kind of a depression in the nuclear membrane. Let's not forget our microtubules. There's some vesicles and fluff associated with this stem. And as this stem gets longer and develops, some microtubules grow between the plates of the stem. Those halves of that stem become separated as microtubule growth continues. Microtubules have, in case you're wondering, mixed polarities in here. In some species, the whole space between is populated by microtubules. In others, it's a kind of a hollow cylinder that develops. Many of these microtubules initially span the whole distance. Recall, this is all going on in a kind of a growing cleft in the nuclear membrane. This bundle of microtubules, hollow cylinder or a solid bundle, is the beginnings of the mitotic spindle. The chromosomes are still inside the nucleus. This microtubule center sort of withers up eventually as the nuclear membrane breaks down and lets that spindle into the nucleus. Now that it's inside the nucleus, this thing, this mitotic apparatus, can interact with chromatin. This object, which has not yet bound any chromatin, is referred to as the central spindle. And these plates at its end radiate other microtubules, and you might guess what their function are. Somewhere in there, there are chromosomes. Which at least initially have some kinetochores on them, or something that looks perhaps like kinetochores. And these chromosomes interact with microtubules coming from this developing spindle, and they, from almost as soon as it enters the nucleus, they get stretched out along it. Until the metaphase diatom spindle looks something like this. Bear with me a minute while I draw it out. So what we have here is these two discs that clearly organize microtubules. The microtubules grow from them. They have mixed polarity, and there's this extensive overlap region in the middle of the central spindle. Lined up on top of that is the chromatin, all of the condensed chromosomes, seemingly stretched out. Now, even to 19th century observers, it was clear that there were pairs of objects in here, and you can imagine what they are. Yes, chromatids joined together, still following DNA replication, sister chromatids joined together, stretched along the spindle, waiting for everybody to get lined up before scission between sister chromatids at anaphase allows them to separate on this apparatus. Now, what do you think of when you see overlapping anti-parallel microtubules? That's right, you think, gosh, maybe they could slide on one another and slide this thing apart. And that's sort of the picture of how the diatom central spindle might work. It captures these chromosomes, lines them up by draping them against the central spindle. They're held together by sister chromatid cohesion until something snips them apart at anaphase, and they allow the central spindle to slide apart. So anaphase looks something like this. With chromosomes liberated to move back toward the poles of the spindle. What I'm trying to depict here is that the overlap, that the microtubules of each half of the central spindle are about as long as they were before, but that the overlap region is much smaller. Ultimately, in telophase, these plates almost seem to wind up into a reforming microtubule center of course, nuclear membranes reform around the mass of chromatin, which decondenses within. Neglected to draw in some non-spindle microtubules there. The spindle itself in telophase disassembles as a cleavage furrow comes in to partition daughter nuclei. And this microtubule center in each daughter is reestablished in association with an interphase nucleus as when we started. All right, let's label these properly. This is obviously interphase. And as this central spindle is developing outside the nucleus, chromosomes are condensing inside the nucleus. This is prophase, during which these poles of the central spindle separate and develop this overlapping microtubule structure between them. Then what is it when the nuclear membrane breaks down? Of course, that's, I should say, if it breaks down. It doesn't break down in all eukaryotes. That's prometaphase. Condensed chromosomes interacting with the new mitotic apparatus. Then we have metaphase. Anaphase. And telophase. It's an interesting thing that at the time the studies that I've summarized in these diagrams are going on in the 70s and before, the view of animal mitosis was actually rather different than it is today. The kinds of ideas people were talking about were not the story that I told in previous lectures, in which kinetochores capture microtubules emanating from spindle poles, stabilize them, develop tension, and then flux through the kinetochore fibers and microtubule motors and things like that, pull separating chromatids, towards the poles of the spindle. Many of those ideas about how kinetochores work and what, how the spindle operates actually came in part from studies of diatoms, which are perhaps not the most tractable experimental subjects in some ways, but those ideas nevertheless informed how we now view animal mitosis, despite the fact that everything looks so very different. The key difference being, of course, that the spindle forms outside the nuclear membrane and is then allowed into it to interact with the chromosomes. Although, a good question to bring to question time would be, is that really very different from what happens in animal cells, where the two sibling centrosomes push off from each other on the outside of the nuclear envelope before the nuclear envelope breaks down and they get to search and capture chromosomes? Okay, at any rate, one of the most obvious similarities with animal cell division for diatoms is that during telophase, there is actually a cleavage furrow pinching down around the separated halves of the mitotic apparatus, and it is in fact supposedly underlain by a slender band of actin filaments. So that looks perhaps not unlike the contractile ring that supposedly powers animal cell division. I don't think anybody knows yet what those actin filaments are doing there, but of course, contracting is a good guess. Now, let's look at how things proceed from here. I'm going to draw out now something much more like a centric diatom in profile. And the main focus here is going to be on how they establish the new valves of the frustrule. Now, I'm exaggerating it quite a bit in this drawing, but the idea is that this hypovalve could fit inside this epivalve, right? And somewhere in here we have the dividing protoplast, that is the actual cytoplasm bound by a plasma membrane. At this point, we would have a nucleus reforming, and the microtubule center migrates around. Let's draw it exaggeratedly large here. Let me put in some girdle bands as well, so that we don't just have a ridiculously naked bit of cell hanging outside there. As the cleavage furrow completes, vesicles gather near the new interface between sister cells, including the Golgi apparatus, the last phase of the secretory machinery. Again, these are meant to be the girdle bands. Think how absurd it would seem to most of our eukaryotic cousins how our cells live without so much as a wall on the outside, just plasma membrane and proteins embedded in it, and maybe a few sugar residues, some sort of a slim glycocalyx separating the plasma membrane from the elements. So the valves here, I'm going to highlight in purple because I've used up all the better colors. Each daughter cell here has to build a new hypovalve. So what is the hypovalve of this daughter here is going to become its epivalve, and it has to build a new one, and that new one is going to fit inside the one that it inherits. Now we have two partitioned daughter cells. Their plasma membranes have separated, they have abscised from one another. The secretory apparatus associated with the microtubule center becomes polarized towards this new cell surface, and a lot of those vesicles fuse into a big sac. These are building what's called the silica deposition vesicle, or SDV, in which the new valves will be secreted and shaped. Oops, remember, they're supposed to fit inside. It is within this silica deposition vesicle that silicic acid taken up from the environment is precipitated to form the new shell. The precipitation takes the form, by the way, of little 30 nanometer balls, typically, that aggregate together. It's hydrated silica, otherwise what we call opal. And think about what has to happen in there. The valves are monolithic, that is, it's a single rock, and a bunch of droplets of stuff are shaped by the vesicle itself into a single fused object. Any structure, any pointy bits sticking out, any holes, any perforations, pores, whatever, has to also be shaped within that vesicle. That is a remarkable constructional feat. Ultimately, the two more or less independent cells, they may be united in a chain or something like that. That's why I say more or less independent. Will secrete the contents of the silica deposition vesicle to the outside world. And it is the secretion of that newly made valve, once it's complete, from the silica deposition vesicle that finishes cell division. With one interesting, essentially universal consequence for these diatoms. And that is, one of these daughter cells is unavoidably smaller than the other because its newly made valve has to fit inside the one that it inherited from its mother, and one of the cells has to get the hypovalve and the other one gets the epivalve. Subsequent cell expansion, incidentally, involves the insertion of girdle bands at this margin to keep from exposing naked protoplasts. That too takes place in a silica deposition vesicle. Similarly, and all of the architecture of the girdle bands similarly must be shaped by the structure of the silica deposition vesicle itself in association with the cytoskeleton.